Howdy, my name is Amelia Couture, and today we're going to be talking about the genetic drivers of immune response in honeybees co-infected with deformed wing virus and sac brood virus. First, we're going to start with introducing co-infection in honeybees. The Apis mellifera, or western honeybee, are important pollinators in natural and agricultural ecosystems, making their health essential for biodiversity. Issues such as co-infection result in detrimental impact on honeybee health and colony survival, leading to possible colony collapse and increasing susceptibility to disease. So what is co-infection? Co-infection is known as the presence of multiple pathogens consisting of viruses, fungi, bacteria, and parasites in honeybee colonies. There are 7 commonly known viruses that can cause severe disease in honeybees that can lead to co-infections. These 7 are acute bee paralysis virus, black queen cell virus, chronic bee paralysis virus, deformed wing virus, Israeli acute paralysis virus, Kashmir bee virus, and sac brood virus. So what exactly is deformed wing virus and sac brood virus in honeybees? Deformed wing virus is a single-stranded RNA virus causing asymptomatic infections such as deformed wings, shorter abdomens, reduced body size, and flight capability, vectored by virodestructors injecting the virus into the bee's hemolymph, increasing its virulence, altering immune gene expression, suppressing antimicrobial peptide expression. Sac brood virus is a non-enveloped RNA virus affecting larvae, causing developmental arrest, characteristic sac-like appearance, and larval death before pupation. Spread through oral transmission, often from nursing bees while feeding larvae. High sac brood levels lead to upregulation of antimicrobial peptides and downregulation of melanization-related genes, suppressing immune defenses. So what genes are activated within deformed wing virus and sac brood virus? Within the RNA interference immune pathway, there are 2 genes activated. Dicer, which produces double-stranded RNA by either replication or byproduct and cleaves the viral double-stranded RNA into small interfering RNA. Arigate-2 loads the small interfering RNA cleaved by Dicer and uses them to target and degrade viral RNA. It's a core component of the RNA-induced silencing complex binding the small interfering RNA and cleaving viral RNA that matches the sequence of the small interfering RNA, destroying viral RNA. Within the JAK-STAT pathways, there is 1 gene activated, which is VAGO, activated downstream of the RNA interference pathway after Dicer2 detects viral double-stranded RNA. Once expressed, VAGO acts as a signaling or messaging molecule activating the JAK-STAT pathway, regulating immune gene expression across the body. So, what immune pathways and genes are activated in response to infections by deformed wing virus and sac brood virus in honeybees? In the study by Durand, adult bees were infected with deformed wing virus A or B and sac brood virus. They were tracked to measure foraging onset, survival time, virus load, and immune gene expression. The results showed that bees that were infected with deformed wing virus and sac brood virus activated the RNA interference pathway, with the genes Dicer and agonite-2 being upregulated, especially in deformed wing virus B and sac brood virus co-infection. The results portrayed that JAK-STAT pathway was activated, including VAGO, which increased significantly with sac brood virus. Here, in a different study, you see a heat map. In this study, honeybee pupae were injected with deformed wing virus A and B, a buffer, and an unmanipulated control, to study the virus load and the immune gene expression. The results exhibited that the RNA interference pathway, including the genes Dicer and arginate-2, had a strong upregulation as virus load increases, particularly from 48 to 192 HPI. In that same study, the results displayed that genes Dicer and arginate-2 that are within the RNA interference pathway showed an increase in expression in the deformed wing virus pupae compared to the buffer injection controls and the unmanipulated controls. This expression rises sharply from 48 HPI, peaking at 192 HPI. The results suggest an early and sustained RNA interference response. So what are the conclusions from these studies? One conclusion is that co-infection in honeybees with infections such as deformed wing virus and sac brood virus cause synergenetic effects, increasing both viral replication and mortality. The timing of infection also matters. Co-infection has more severe effects than sequential ones. The gene immune system shows heightened responses, particularly through the RNA interference pathway, with it being the main immune defense activated in response to deformed wing virus. The magnitude and timing of an immune gene activation corresponds to the viral load of the infection. There is a significance to these studies. These studies demonstrate that deformed wing virus and sac brood virus alone are less harmful as when they occur together. Colony decline is not due to a single virus, but synergenetic interactions between viruses. The studies also portrayed how different immune pathways are activated, such as RNA interference and JAK-STAT, depending on the virus and infection route. RNA interference being the key immune pathway activated with deformed wing virus. Co-infections with deformed wing virus and sac brood virus result in accelerated viral replication, increased immune system activation, and higher early mortality rates. Here are the references used for this study.